This is session 8 on applied amphibian conservation. So yeah, once again, thank you for sticking around. And I'm very excited to introduce Barbara Beasley, who will be telling us about her work with underpasses and fencing and road mitigation otherwise, and work with the northern red-legged frog. So when you're ready, Barbara, please share your screen and take us away.
Okay, thank you very much. I want to thank all the conference organizers and everyone who's still here participating. I'm really glad for the opportunity to learn and be inspired by everyone's work and to share what we've been doing. I work with a small nonprofit on the west coast of Vancouver Island, and we do work that promotes stewardship of amphibian habitats through research and educational projects and public outreach. And a lot of our work is focused on addressing the threat of roads. Whoops. Sorry. And we affectionately call this work the SPLAT project, and we put out an annual newsletter, and I want to share some of what's in the update of that newsletter with you by telling you about a variety of things that we've made happen in terms of mitigation and the kinds of monitoring that we've done or in the process of doing or would like to do in the future to test the effectiveness of different mitigations for reducing road mortality and connecting habitats. We really want to make sure that the structures that we've helped put in place are beneficial. And this weighs really heavily on my mind, especially as I've become more and more aware that some structures that we assume are helpful could actually be doing more harm than good. So a cautionary tale comes from this study that was done in the Netherlands, where they showed that fencing actually served as a barrier that they attributed to a decline in a breeding population of toads. And that was certainly not the intention of the mitigation. But it was believed that the underpasses that were put in place were too far apart for the toads to be able to find them. And it's really easy to see how this can happen. As Cheryl Brame, who has done a lot of research to inform California's best management practices, stated, there's very little research available to guide decisions about the number of crossings and the spacing between crossings that's required by different species. So different species have really different needs. And I would add that there are many other decisions related to the features of mitigation structures that we really don't have good information for making decisions about. So I know that I'm preaching to the choir when I say we need more effectiveness in monitoring and research. I've been inspired and learned a lot from the work that many of you have done and published in the past. And I'm super grateful to CHS for having hosted Tom Langton's talk earlier in the year. I found it tremendously helpful and inspirational. So I want to tell you about our work. We are on the west coast of Vancouver Island, and we work within the traditional territories of the Nuu-chah-nulth. And specifically, I work within the project I'm going to tell you about and where I live is within the traditional territory of the Uklilot, who have stewarded the area since time immemorial. The highway that I work on connects over 1 million visitors a year to Tofino, Ucluelet, and Pacific Rim National Park Reserve, which has beautiful surf beaches. The highway, like many in Canada, bisects habitats where amphibians are crossing and being killed in pretty high numbers. And the two places that I work on where the highest densities of road mortality occur are shown with the 2 stars on the map. And at these sites, we have 4 aquatic breeding species that are crossing when they disperse or migrate between breeding habitats and non-breeding habitats. And so they are the Rana, Aurora, the red-legged frog, Ambystoma gracili, the northwestern salamander, Pseudocris rigilla, the Pacific tree frog, and Tarica granulosa, the rough-skinned newt. And in addition to those 4, which are the ones predominantly seen on the road, we also have 2 plethodontid salamanders. So the western red-backed salamander and the wandering salamander. And what they're doing on the road is anybody's guess, but I imagine they're feeding or looking for mates, or maybe just trying to escape from some water-saturated soils during some of our torrential rainstorms. So in order to address the issues of mortality and connectivity, We have been involved in helping Ministry of Transportation and Infrastructure and Parks Canada install some box culverts, 4 new concrete box culverts at peak crossing points that are near Swan Lake. One went in 2011, the other 3 went in just last year. We've also installed fabric fencing that angles from the forest to the entranceways of the new culverts. And we've also installed fencing to 3 existing metal drainage culverts along a stretch. And the stretch is shown there on the road. Those are, the black lines indicate 15-meter segments to give you some sense of what's going on. The triangles are hydro poles that we use as markers to help find our way. And then the fencing is indicated as shown in the legend. Interestingly enough, last year when the Parks Canada installed their 3 box culverts, they did so in order to mitigate the effects of a bike path that they put right next to the highway in this peak crossing area. And when they did that, they actually constructed the trail on top of lock block wall. And we then got them to modify the lock block wall so that it's covered with a a root barrier plastic, and then has like a metal strip of aluminum that's folded over to create an overhanging lip that hopefully will prevent amphibians from accessing the path and the road. But quite a bunch of different sets of mitigation, and kind of like finally pretty good sample size for monitoring the efficacy or the effectiveness of these different kinds of structures. Okay, then at the Lost Shoe site, this is a different kind of a site. It's a bridge, and amphibians are moving along a riparian zone along the creek that then goes under the bridge. We installed fencing to keep amphibians off the road at this site and guide them to the bridge. And last year, we were quite worried about the bridge itself because it's covered, the banks of the bridge are covered in riprap, and that armors the banks so that they won't erode. And that's standard practice from the Ministry of Transportation here in BC and probably in lots of other jurisdictions as well. But anyway, we have observed some of the red-legged frogs having a hard time crawling across that riprap, sometimes getting stuck, it looked like, in some of the crevices. And so we decided to cover the riprap with finer materials, and then we added soil, plants, and cover objects to make a more amphibian-friendly path. So the question is, and these are ongoing questions, how well do these structures work? So I just want to give you a really brief overview of kinds of the approaches we've taken, and really a sort of a snapshot of some of the results. So let's start with the fencing. We started by testing different kinds of materials as barriers for the 4 aquatic breeding species that we were finding crossing the road. And we used experimental enclosures made out of different materials. So you can see the figure here shows the 4 different kinds of materials we tried. And then we would put animals in them, and we would use a camera to see what happened overnight, see whether they could escape. And we did find an effect. We found that the mesh, which is like a plastic aqua mesh, was very easy for especially Pacific tree frogs to climb, so they could escape from it. The other 3 didn't really differ that much, so any of them would seem suitable as potential barriers for this suite of species that we have. Okay, then the second thing we did was we looked to see whether or not amphibians were escaping from the angled fencing that we'd placed to the entranceways of the culverts and getting up to the road. And we did that by putting sort of a back catcher set of fencing along the roadside. So if you look in the bottom left corner, you can see the arrows. So what we're looking to see is how many of the animals that we marked along the fences that angled, how many of them actually were then recaptured along the roadside fences. And we could say that very few actually made it to the roadside fences. And over time, we actually saw fewer and fewer animals, even unmarked animals, getting there. So we felt pretty confident that those angled fences were working well. Okay, let's go over to the Lost Shoe, where we also installed some fencing and where we wanted to find out whether that was working. So we did that by looking at road mortality. So we had done road surveys both before and after we installed the fencing. And we looked to see what the mortality was like in sections that were fenced and unfenced. So a a batchy design. Now, we only did it for 1 year before versus 1 year after, although we have repeated the study last year, but I'm not showing the results for that. But here you can see that we did have quite a reduction in the number of roadkill at the fenced sections. And especially red-legged frogs, we saw quite a significant reduction and not that much difference between the years in terms of what was moving across the unfenced sections. So we quite like that result. When we did the study again last year, we did see a little bit more mortality in the fenced sections, and that helped us figure out where to go and do some patching along the fencing that had deteriorated since we'd installed it in 2014, or sorry, in 2013. So then the other question we wanted to figure out was how well did the structures we put in work for connectivity? And so to do this, we monitored movements along the fences and through the culverts, and we used a variety of different techniques. We tried using fluorescent powder. It didn't work very well, but then we used mark recapture using visible implant elastomers and pit tags. And then we also used camera traps. So with the mark recapture, we would try to capture the animals as they were coming out of the culvert on the opposite side of the road to see how many of the ones we'd marked on one side were actually recaptured on the other side. And we used what we called our exit traps. But I just wanted to highlight that not all animals made it into the exit traps. There was a little gap so that they could move along the fences and avoid the exit trap. So if you look at the graphs on the bottom, you'll see that we did see quite a number of animals moving through, especially when we used our camera traps. So the green bars show the ones that were photographed in both the box culverts and the existing drainage round culverts. And so this is just a sample of one culvert of each type. We didn't have very much recapture though. We recaught very few of the marked individuals on the opposite side of the road. So there's some issues with the techniques, I think. One was that we weren't necessarily able to capture everything that went through. And the other is that the photographs that we got were not always the greatest. But I want to show you that, yes, indeed, we did see animals moving through both types. So I'm going to give you a bit of a slide presentation through some of our time-lapse photos. So take a look and try to find the eye shine as these frogs are coming through the box culvert. This was a big night in 2012, April, the 25th of April. We saw quite a few lovely frogs coming through. And then here's another, this is the round culvert, the drainage culvert. It doesn't always carry water, so it is quite suitable. And here you can see a couple of animals that are moving through. So that's fun. We also then were looking at, if I'll remind you that at the Lost Shoe site, we have a bridge that was covered with riprap that we thought was a problem. So last year, we decided to cover the riprap with finer materials, and we used camera traps to see whether or not we saw a higher frequency of animals moving under it after the treatment. And I've circled the results. We did indeed find 2 to 3 times the frequency of red-legged frogs and salamanders moving underneath underneath that bridge after we had done the treatment. But there's a caveat, of course. It's probably easier to detect them on a smooth path than it is on riprap. And unfortunately, we didn't think of that until later. Hindsight is 2020. So we're still racking our brains to try and figure out how we could maybe look at detectability questions. So any ideas would be much appreciated. Okay, the other thing about the Lost Shoe site is that we have these great long 100-meter fences that are parallel to the roadside. And we really don't think that amphibians are following them all the way along to the entrance of the bridge. On the left side here, try and find the newt, which is crawling along the fence in the direction of the bridge. That's an amazing sighting because we rarely saw that. Usually they just sit there and stare at the fence. And we also found very few matching nights when we saw them on the fence and going underneath the bridge. So we really don't know. I mean, it's hard to know whether we're detecting them very well, but we don't know whether they're actually following these structures. And then finally, the big question is, well, are we doing more harm than good? Well, we need kind of an ultimate proxy measure of whether or not we're doing any good. And we want to know whether or not our structures are actually maintaining the population. And so we do, and we have been doing egg mass surveys for red-legged frogs and northwestern salamanders. We've been doing them since 2008, but we did change our methodology in 2014 to using stand-up paddleboards instead of kayaks. And we're still trying to figure out how to correct some of our earlier measurements with a kayak, because it's much easier to detect them with SUPs. So anyway, to summarize, we do have a pretty good handle on the fencing. We can make it work for barriers, and we feel pretty good about that in terms of the barrier function and reducing road mortality. We do see animals, especially our target species, moving through the culverts and the bridges, maybe not in as high numbers as you'd expect as those given along the guiding fences, but they certainly are still moving through them. We still maintain that the dirt helps at the bridge, but we would like to figure out the detectability issue. And then we really want to learn how to improve the fencing. We want it to be a good guiding structure. We're wondering whether angled configuration works better than the ones that are parallel to the road. We're also interested in figuring out if there's ways we can put little back loops or additions to the fencing to get them going in the right direction. And finally, this whole bike path, this whole bigger system that's been put in place, we really want to monitor it, find out if it's working for the park. So that brings me to my next slide, which is we're looking for fresh ideas and energy to help us out in all kinds of ways, helping us with study design, helping us with advice for data analysis. We'd love it if anybody's got grad students and wants to supervise them in our neck of the woods. And we also appreciate any encouragement in helping us get off our butts and publish our results. So if you want to be involved, please let me know. I just want to finish off with our acknowledgements, lots of different funding partners and lots of support locally and within British Columbia and from the government, and lots of great students and volunteers, lots of community volunteers. So thanks.
And next we will invite Briar Hunter up to talk about her work in captive and wild Oregon spotted frogs.
All right, so yeah, I'm Briar Hunter. I'm a master's student through Laurentian University and the Toronto Zoo. And I'm gonna give you some of the background on some of my research so far and ultimately tell you how 3 zoos, 3 organizations, and 1 university lab have come together to buy time for Canada's most endangered amphibian species, the Oregon spotted frog. So the dashing frog that you see here before you has disappeared from up to 90% of its historical range. It's reduced to only 6 populations left in all of Canada, and it's estimated that fewer than 700 adults remain in the wild. This is a frighteningly small number for a frog species. So, as assurance against extinction, and in order to breed tadpoles to release to the wild, 3 captive populations were established back in 2009. They're at the Vancouver Aquarium, the Greater Vancouver Zoo, and the Toronto Zoo. Unfortunately, all 3 of these facilities are experiencing low reproductive output. A potential culprit for this is genetics. Retaining genetic diversity is crucial in captive populations as it affects not only reproductive output, but also offspring survival. Due to their small and fragmented populations, we don't know if these frogs were genetically healthy before they even entered captivity. So I'm asking a couple questions. The first is whether current breeding methods are retaining sufficient genetic diversity to be sustainable long-term. And is inbreeding higher in captivity than in the wild? And do we see differences between our zoos with their slightly differing breeding strategies? To tackle these questions, I've collected DNA from all of our captive frogs using buccal swabbing. And I sought a sample size of 30 from each of our wild populations. Unsurprisingly, I fell short of this goal at some of our more elusive sites, but we're working with what we've got. Right now, all of my swabs are waiting to have the DNA extracted, amplified, and sequenced. I'm planning to analyze them using ddRAD-seq, which is just a sequencing method, depending on the quantity and quality of DNA we extract. From here, I'll ultimately be comparing the genetics of our captive versus our wild populations. to assess their genetic diversity using the proportion of polymorphism and average heterozygosity, which are just two different measures of variability, which is something we want to see in our populations. And the inbreeding, looking at both the population and individual level, using that observed to expected heterozygosity, which is something we don't want to see in our populations. I don't have any results to share with you yet, so I'm going to tell you my prediction. Overall, I'm expecting to see low genetic diversity across the bar, with our lowest levels in those captive populations. Conversely, we expect to see high inbreeding levels, with those highest coefficients in our captive population. Although we do expect our zoo breeding strategies are helping to retain genetic diversity, if there's inbreeding in the wild, we're going to see it in captivity as well. Let me add a small caveat to this. Inbreeding is not the same as inbreeding depression. The former, which is what I'm measuring, merely tells us whether related individuals are mating more than expected, aka just how much incest is going on here. On the other hand, inbreeding depression is that reduced fitness, for example, in offspring survival. If we find high inbreeding levels in our populations, we can then take the next step and assess whether inbreeding depression is happening. However, this past spring, one of our facilities had the most successful breeding season we've ever seen in captivity, producing and releasing more tadpoles than the previous 9 years combined. This exciting accomplishment would suggest that inbreeding depression is not the main culprit of that historic low reproductive output, but it could still be a factor. I look forward to updating you next year. In the end, this study will inform on the current and long-term sustainability of these captive breeding programs and provide insight into these reproductive issues and whether they can be resolved. Overall, I'm hoping to improve the cohesiveness of Oregon spotted frog recovery efforts across Canada to ensure that all of our time, resources, and efforts are making a lasting impact on this endangered species. As I mentioned, there are a ton of partners and people involved in this project in Ontario and BC, and so a big shout out to all of them, and one final one to all of you for listening. Thanks.
Bonjour, rebonjour. Ça me fait plaisir de présenter un autre projet de conservation sur une espèce qui nous est chère et qui est notamment étudiée en captivité. Et nous allons vous parler à Aurore Fayard à propos du développement larvaire chez la rainette faux-grillon.
Bonjour à tous, welcome everyone. My name is Aurora, and I'm just before switching to French, I just wanted to encourage everybody to ask me questions, even in English. I will be happy to answer them. Donc bonjour à tous, moi c'est Aurore, je travaille à l'Université Laval pour le laboratoire de Marc Mazerolles en co-direction avec Vance Trudeau, puis j'aimerais vous présenter aujourd'hui notre travail sur le développement larvaire chez la rainette faux-grillon. Donc cette espèce malheureusement est victime de la dégradation de son habitat et de la destruction de son habitat. Donc on a perdu 90% de son aire de répartition historique. Vous pouvez la voir sur la carte ici en quadrillé. Puis en vert, c'est son aire de répartition actuelle. Donc la rainette faux-grillon est présente dans 2 régions du Québec, l'Outaouais et la Montérégie. Donc moi, je travaille sur les populations de Montérégie. Et en 10 ans, on a vu 1/3 des sites de reproduction répertoriés qui avaient été détruits. Donc c'est une situation assez inquiétante. C'est seulement les populations qu'on appelle du Grand Lac Saint-Laurent et du Bouclier canadien qui sont protégées par une loi fédérale et une loi provinciale. Puis, en fait, mon projet de maîtrise répond directement à un des objectifs du plan de conservation qui a été mis en place face à ce déclin, face à cette situation, qui est de développer des connaissances et des techniques pour l'introduction d'individus et établir de nouvelles populations, en d'autres mots, une réintroduction en nature. Donc, on a publié récemment un papier avec mon collaborateur qui va vous parler juste après, Jeffrey, qui fait le résumé en fait des connaissances qu'on a dans le genre Pseudacris, des 18 espèces qui existent. Et effectivement, la rainette faux-grillon fait partie de ces espèces qui en fait, on a peu de données sur son cycle de vie, sur la dynamique des populations. Pour vous donner un exemple, on n'a pas d'estimation exacte du reste des adultes qu'il y a dans les populations les dernières populations au Québec, donc ça pose problème pour sa conservation. Je vous ai mis ici le cycle de vie pour juste indiquer que ces espèces ont des cycles de vie qui sont courts. Et on a aussi encore des données qui divergent, par exemple, sur la longévité, donc des éléments qui manquent. Ensuite, pour le contexte, j'aimerais dire qu'on a travaillé avec le Biodôme de Montréal. Donc, en fait, ils sont occupés de la partie captivité, de la reproduction en captivité. Donc, on a collecté certains adultes cet été, puis on a pu avoir une reproduction dans leurs locaux avec injection d'hormones, qui a stimulé la reproduction, avec l'Université d'Ottawa qui a fait ce cocktail magique. Puis on a eu beaucoup d'œufs qui ont éclos. En fait, on a eu plus de 2000 têtards qui ont réussi à survivre donc dans ce genre de bac que vous voyez en blanc. Donc certains de ces têtards ont été utilisés dans l'expérience que je vais vous montrer aujourd'hui. Et certains se sont restés en captivité au biodôme jusqu'au but ultime qui était la réintroduction dans des étangs qui sont aménagés depuis plusieurs années pour justement la rainette faux-grillon et pour sa réintroduction. Donc ici, j'ai mis une photo d'un des étangs naturels, en fait, là où vivent les rainettes faux-grillons encore aujourd'hui. Donc c'est des zones humides temporaires. On a une densité de larves qui va varier selon le niveau de l'eau. Ce qui nous intéresse, c'est de savoir justement avant la réintroduction, on voudrait évaluer l'effet de la densité et de la présence de litière forestière dans ces environnements sur le développement larvaire. Puis le deuxième objectif, c'est d'estimer ce qui nous manque, les taux de survie qui vont du stade larvaire jusqu'à la métamorphose, puisque aujourd'hui on a très peu de données sur ces survies-là. Donc, on a émis les hypothèses suivantes, c'est qu'à basse densité, donc une densité de têtards, on a un meilleur développement comparé à une densité qui est plus haute. Ici, on a choisi la densité 0,1 têtard par litre qui est trouvée dans la littérature pour cette espèce dans le milieu naturel et 1 têtard par litre qui fait plus référence à une densité qu'on trouve en laboratoire, par exemple. Ensuite, la présence de litière forestière, on pense qu'elle va être bénéfique au développement larvaire, même si vous voyez ici, la canopée est ouverte et c'est une espèce qui utilise plutôt des espaces ouverts. On va sûrement avoir des débris de feuilles ou quoi que ce soit qui va s'accumuler. Puis la troisième hypothèse, c'est que la survie à la métamorphose va être meilleure quand on a une densité qui est plus basse qu'à une grande densité. Donc ce qu'on a utilisé, peut-être que certains d'entre vous sont familiers avec cette méthode, on a choisi d'utiliser des mésocosmes, donc c'est des dispositifs communément utilisés pour les amphibiens. Il permet de recréer les conditions des milieux naturels et tout en gardant en fait un contrôle sur certains paramètres qui sont importants pour nos études. Donc ici, on a choisi un bloc complet aléatoire, 12 mésocosmes qui sont disposés aléatoirement dans l'espace. Puis on a donc 3 réplicas grâce à ça. Donc, 4 traitements qu'on a testés à faible densité. Donc, comme j'ai dit, 0,1 têtard par litre, on va le tester sans feuilles et avec feuilles. Donc, ça va être nos 2 premiers traitements. Puis, les 2 autres, ça va être une densité 10 fois supérieure, donc 1 têtard par litre sans feuilles et avec feuilles. Donc, vous voyez à quoi ça ressemble ici sur la photo en vrai, avant remplissage. On a choisi d'utiliser 1 litre d'eau qui venait des étangs aménagés, donc exprès pour sa réintroduction. On a filtré ce 1 litre et on l'a mis dans chacun des 12 mésocosmes, dont chacun a eu 1 litre, bien sûr. Et le reste, on l'a rempli avec de l'eau plate. Donc, finalement, on a un total de 180 litres dans ces mésocosmes, ce qui va nous permettre d'avoir 18 têtards dans la densité 0,1, logiquement, et 180 dans les bacs avec une haute densité. On a aussi ajouté 30 grammes de plantes aquatiques dans chaque mésocosme. Et pour ceux qui avaient le traitement avec feuilles, on a ajouté 100 grammes de feuilles d'érable et de bouleau. Donc, on a évité le hêtre et le nerpin cathartique, par exemple, qui peuvent être des feuilles avec un effet toxique ou néfaste pour les têtards. Donc, on a attendu 1 mois. 1 mois après, on a pu faire le transfert donc, du biodôme de Montréal, là où les têtards avaient été élevés, jusqu'au parc national du Mont-Saint-Bruno, là où les étangs et les mésocosmes sont. Puis, finalement, avec un rapide calcul, là, on a transféré 1188 têtards dans ces mésocosmes. Et il y en a une partie, donc à peu près la moitié, un peu moins qui sont restés au biodôme pour la captivité. Voilà à quoi ça ressemblait. On a dû protéger les mésocosmes contre les prédateurs. Donc, c'est ce grillage à poules au-dessus en gris, c'est pour protéger contre, par exemple, les ratons laveurs qui pourraient rôder dans le coin. On a ajouté des moustiquaires pour excusez-moi, pour que la qualité de l'eau ne soit pas altérée, qu'on n'ait pas des insectes qui viennent aussi pendre leurs os. Et aussi, on a ajouté beaucoup de morceaux en fait, de liège qui permettent d'être des flotteurs et qui facilitent la sortie, l'émergence des métamorphes. Vous voyez par exemple ici sur une photo, on avait utilisé des clôtures par-dessus ces flotteurs et les rainettes faux-grillons métamorphes pouvaient vraiment sortir facilement. Donc, on venait tous les 2 à 3 jours suivre le pH et la température de chaque bac, le niveau de l'eau, on voulait qu'il reste à la même hauteur puisqu'on teste ici la densité. Et on a aussi nourri les têtards avec du phytoplancton et du zooplancton. Puis ensuite, une fois qu'ils étaient sortis, ces petits métamorphes, on a attendu que leur queue se résorbe entièrement et on a mesuré et pesé chaque individu. Donc, pour les résultats, on voit ici un graphique de la température moyenne des bacs en fonction de la date. Donc, on voit que l'émergence, elle a commencé environ vers le 9 juin. Les bacs étaient assez chauds, donc 25 degrés en maximum, et ça a fluctué jusqu'à 17,5 degrés. Donc, ici, vous voyez un graphique qui présente en fait les différents traitements, le nombre d'émergences des individus par jour, parce qu'on venait, une fois qu'on a eu le premier métamorphe, on venait tous les jours regarder dans chaque bac et collecter les individus qu'il y avait. Donc, le pic d'émergence a quand même duré longtemps, 3 semaines environ. Et on a eu entre 20 et 40 individus par jour. Et ce qui est intéressant de voir sur ce graphique, c'est que les premiers jours d'émergence, les 3 premiers jours, on voit une couleur rouge et une couleur dorée. Et ça, c'est des traitements de densité, donc 0, 1 et 1, mais avec de la litière forestière. Donc, on a peut-être un effet précoce des traitements qui avaient en fait des feuilles mortes, donc ce qui peut être expliqué par un apport de ressources supérieur à ceux qui n'avaient pas de feuilles, par exemple. Mais ça, ce n'est pas mesurable statistiquement, c'est juste observable ici. Donc voilà, ça a fini à peu près mi-juillet. Puis ensuite, on a fait des analyses pour répondre à nos questions. Donc, une analyse de variance, vous voyez ici le taux de survie en fonction des traitements. On ne voit pas de différence significative de taux de survie, en fait. C'est-à-dire qu'ils ont tous les traitements, dans tous les traitements, il y a eu une survie globale de 46%. Donc, en tout, on a eu 542 émergences sur le total réintroduit. Donc, c'est ce qu'on trouve à peu près dans la littérature. C'est dans la moyenne de la survie qu'on trouve aussi dans la littérature. Donc, c'est une bonne survie, mais c'est juste qu'il n'y a pas eu de différence entre les traitements. Ensuite, on voit ici une relation claire entre la taille de l'individu puis sa masse. Donc, c'est corrélé, c'est-à-dire qu'un individu qui est grand aura une masse plus importante. Mais on voit que la plupart des individus qu'on a mesurés, donc là, c'est tout traitement confondu, ils faisaient de 8 à 9 mm, donc c'est très petit, hein, ça fait moins d'un centimètre, puis entre 50 et 100 mg. Donc, on a mis en place, on a essayé de créer des modèles linéaires mixtes avec des effets aléatoires pour prendre en compte le débalancement des données, puisque bien sûr, il y avait moins d'individus émergents sur des bacs où il y avait 18 têtards versus 180. Donc, on a fait des contrastes pour essayer de répondre à nos questions. Donc, un contraste A, B et C. Donc, j'ai représenté ça avec des lettres sur les graphiques. Donc, on voit ici que la taille, qui est la variable réponse ici, en fait, on a le meilleur traitement où les individus étaient les plus grands, c'était le traitement la basse densité, donc 0,1 tétard par litre avec feuilles. Et il y a un effet significatif des feuilles, donc un bénéfice finalement des feuilles, puisque c'est significatif à 1 étoile avec la même densité mais sans feuilles. Puis on a fait pareil, on a essayé de savoir s'il y avait une différence de taille avec la même densité avec et sans feuilles, puis il n'y en avait pas. Par contre, ce qui est vraiment intéressant, c'est que si on compare les densités maintenant, donc avec le contraste C, on a comparé donc le groupe ici avec 0,1 avec et sans feuilles, puis le groupe 1 tétard par litre avec et sans feuilles. Puis il y a vraiment une différence, c'est-à-dire que les individus sont globalement plus grands lorsqu'ils étaient seulement 18 versus 180. Donc, on a fait pareil pour la masse. On a les mêmes effets qui reviennent, en fait, une différence encore plus significative ici pour le groupe à basse densité. Donc, les feuilles sont bénéfiques aussi pour avoir une masse importante. Puis, c'est aussi ce traitement-là qui est le meilleur et qui donne des individus qui sont quand même assez corpulents. Donc, je voulais juste faire une discussion sur les têtards en fait, qui sont restés au biodôme en captivité. Malheureusement, on a vu beaucoup de métamorphes mourir en captivité. Donc, il y en a beaucoup plus qui sont morts versus dans les mésocosmes. Donc, il y a eu 79% de mortalité et beaucoup sont morts en fait après la métamorphose. Donc, on voit sur les photos, il y a eu des problèmes notamment de motricité, des organes qui étaient dysfonctionnels, la morphologie aussi de la bouche pour celui-là et des rétentions d'eau ou d'air. Donc, on a soumis certains individus pour comprendre un peu ce phénomène puisque c'était les mêmes pontes finalement qui venaient. Enfin, c'était les mêmes parents, donc les mêmes têtards que moi j'ai pris pour mon expérience et pourtant j'ai vu que l'état des têtards était meilleur, enfin des métamorphes plutôt étaient meilleurs dans les mésocosmes. Donc on en a soumis certains au Centre québécois de la santé sur les animaux sauvages et malheureusement on n'a pas eu de réponse là, de maladies significatives ou d'éléments qui nous auraient éclairés. Donc on attend toujours des réponses de qu'est-ce qui se serait passé en captivité. Bon, pour revenir aux hypothèses, on a pu conclure qu'à basse densité de têtards, on avait un meilleur développement comparé à une haute densité, donc toujours 0,1 versus 1 têtard par litre, et que la présence de litière forestière était bénéfique au développement larvaire lorsqu'on avait peu de têtards dans les bacs. Donc là, on n'a pas vérifié ça pour la grande densité. Puis, on n'a pas trouvé de différence significative entre la survie à la métamorphose entre les différents traitements, mais c'est quand même très important de surligner que pour une réintroduction, c'est quand même des résultats intéressants parce que ça veut dire que des individus qui viennent, qu'on met avec une faible densité, auront une meilleure fitness par la suite. Par exemple, on sait que la survie des juvéniles est dépendante de la taille à la métamorphose. Donc, on sait aussi qu'il y a une corrélation possible entre la taille de la femelle et le nombre d'œufs qu'elle pond. Donc, ça, c'est des éléments vraiment importants quand on parle d'une réintroduction. On veut maximiser la survie, mais on veut aussi maximiser le futur des nouvelles populations qu'on introduit. Donc, les messages à garder en tête, c'est que ce dispositif a été efficace pour élever des larves de rainettes faux-grillons et que les conditions optimales étaient une faible densité et ajout de litière forestière pour maximiser la fitness pour les nouvelles populations à venir. Donc, je tiens à remercier vous d'être toujours là et un grand merci à mon équipe de terrain cet été et aux partenaires financiers qui nous soutiennent dans ce projet. Merci beaucoup.
Thank you very much. And our last speaker is Jeffrey, and his topic will be combining very nicely our two most recent topics. We'll be enjoying both discussions about the chorus frog and captive breeding. So thanks very much, Jeffrey. We welcome you up to take us home here at CHS 2021.
So I'll get on with it. So yes, my name is Jeffrey Ethier. I'm a PhD student at the University of Ottawa, and I'm going to talk a little bit more about the collaboration that Aurore set us up with about using a collaborative approach to the captive breeding and reintroduction of chorus frogs. So as Aurore said, Western chorus frogs are currently declining in Ontario and Quebec, and they have many threats, including human disturbances and potentially climate change. These frogs use temporary water, which is why climate change and human disturbances changing the hydro period of these temporary water systems are extremely detrimental to the species. Currently, there's a lot of initiatives in the Montérégie region of Quebec for the protection and habitat creation. However, recently we're being initiated captive breeding and reintroduction methods to bolster the populations that are declining. And this relies on several institutions, not just for their on-the-field help, but also their experience throughout the years of this species. And that includes both people in Quebec as well as myself and my collaborators at the University of Ottawa to research methods for captive breeding and reintroduction. So as Aurore also said, and she covered very well, we collect individuals from several populations in the Montérégie region. We then bring these adult frogs to the Montreal biodome to induce spawning via hormones because this species does not breed very well in captivity. After that, they are taken to these mesocosms where Aurore and her colleagues did some experiments on larval development. So they were looking at emergent rate, size and mass and metamorphosis, as well as the effect of leaf litter on them. And she's already gone over this in a lot of detail, so I won't spend any more time there. So what I'm doing at the University of Ottawa is really investigating how we can improve the captive breeding scenario. So even with hormones, these species do not breed all that well, or there's high variability. So some individuals will have a large amount of eggs produced, some individuals will only produce very few. So I'm using individuals that are from Ontario to infer information about the population in Quebec. So first I'm going to look at calling differences between the two populations, because I believe that the main element that is missing from captive breeding is this acoustic environment that the frogs would be used to in their natural environment. So usually these frogs gather in very early spring or late winter, and like hundreds of individuals will be calling. And I think this is a real key element for the hormone stimulation of reproduction, which is currently absent in a captive breeding environment. You can just imagine a bunch of frogs in tanks, and there's not that same effect of hundreds of individuals gathering and calling. Second, I'm going to look at the dose that we use for the hormone injections and see, does this affect calling behavior? Then I'm going to see if there's various elements of the properties of the acoustics that affect the fertilization rate when these couples breed. And finally, I'm going to see if playbacks of males calling can influence the amount of fertilization rate in eggs, as well as the production of eggs, so the number of eggs that are produced. So the overall significances of this is that amphibians are one of the most endangered taxa, with over 40% of species declining across the world. The information that we gather would be transferable to any species that use acoustics in a captive for reproduction, and then transfer these to captive breeding scenarios. And I do believe that the key element to the success of this project is the amount of collaboration. And overall, it's important not only to ask these questions and infer this information for species that are in serious decline and are very, very low abundance, but also in species such as the chorus frog, which are currently still relatively abundant, but steeply declining, with the overall goal of keeping common species common. Thank you.
Bien que je sais que Patrick parle très bien français, je vais quand même couvrir les questions qui sont dans le chat. Donc pour Aurore, une des premières questions de Jonathan, c'était: est-ce que tu connais le taux de mortalité dans tes mésocosmes?
Oui, finalement, c'était un taux global, comme j'ai montré, c'était 46% de survie, donc on a un 54% de mortalité, versus on a eu 79% de mortalité quand on était en captivité. Donc ceux qui sont restés au biodôme ont eu plus de mortalité. Mais moi, j'ai eu à peu près 54%, donc plus de la moitié.
Est-ce que les 79% au biodôme, c'est une moyenne sur plusieurs années, ou il y a des années qui sont beaucoup mieux que ça?
Non, et ça, c'était la première année qu'on réessayait, bien que le biodôme fait ça depuis plusieurs années consécutives. Il y a eu une réintroduction déjà avec le biodôme qui a été faite. Donc, ils ont l'habitude de prendre soin des têtards. C'est vraiment cette année qu'on a vu une grande mortalité qu'ils n'avaient pas eu les années précédentes. Donc, c'est quelque chose qu'on ne comprend pas encore. Puis pourtant, il n'y a pas eu d'infection à la chytridiomycose, qu'on aurait cru être un facteur possible, mais les métamorphes n'étaient pas positifs au BD. Donc, il y avait des adultes qui l'avaient été, mais eux ne l'étaient pas. Donc, c'est toujours un mystère.
Je pense qu'il y a beaucoup de choses en parallèle à discuter avec le projet sur la grenouille de l'Oregon, parce qu'eux aussi, ils ont eu
Oui.
beaucoup de mortalité, sauf l'année dernière, et on n'en est toujours pas trop sûr pourquoi. Une autre question pour toi, de Amanda. Est-ce que tu penses que la litière pourrait aussi fournir un environnement moins stressant pour les animaux qui puissent se cacher, voire thermoréguler quand tu places de la litière?
Bah oui, clairement, ça peut vraiment avoir cet effet-là, c'est-à-dire que ça peut être juste un refuge parce qu'ils sont exposés à la lumière, il y a rien d'autre finalement que les morceaux de liège qui puissent se cacher en dessous. Donc on a vu souvent, c'est ça, que quand on regardait les têtards grandir, on les voyait pas souvent finalement dans les bacs, ils étaient souvent cachés. Donc ça, c'est très possible, oui. C'est une très bonne remarque.
There's a question for Jeffrey this time. Do you think there's a link in male calling and sperm fitness that may cause higher fertilization rates?
Yeah, I kind of breezed over that just because I had the 5 minutes. But yes, that's what we're expecting, is some sort of correlation between sperm fitness and the calling properties and resulting in higher fertilization.
Do you think the actual acoustic tests have more impact on male or female frogs?
That's another great question, and I'm trying to brainstorm how I can incorporate that with my current proposed research, and I'm really thinking really hard about that. Mainly, I think it is the effect on the females, but I'm trying to think that maybe there's also a role in male-to-male competition.
Abnormalities. Pam, would you mind to precise more what abnormalities you're looking for?
Yes, yeah, if some of the abnormalities that Aurore spoke about that were showing up, is that related to sperm quality, or that would be just more housing conditions?
I can answer on my end. We don't really know, as Aurore said, we don't really know what the exact cause of it. It might be the housing conditions. It might also be just the food variety. We only are able to feed certain number of prey species to them. That might be effect. It might just be, this is something that occurs in nature that we just don't see because they die and they dry up so quickly. So yeah, we're still scratching our heads a little bit on that one.
Nick.
Hi, yeah, I just continuation on the questions for Jeffrey. I'm just wondering what you think of in terms of, is it individual call or is it chorus that you're really focused on? Like, are you're trying to quantify individual males on their call, but are you playing them in a chorus or are you playing them just on their own?
So I'm looking at individual variation and fertilization rate, but for my experiment where I play back, it will be like a chorus from either a local pond where the individuals that are collected for the Montreal biodome breeding, and also to compare that to an Ontario course to see if is there an effect on the population? Does one population cause an effect in the fertilization rate? Is there, to kind of look at individual variation between, not individual, sorry, population variation in calling behavior.
Okay, what sort of metrics are you looking for in your individual variation between, are you looking for temporal
I'm looking
elements,
at a whole,
like species recognition, or are you looking for pitch and tone, more quality?
So there's a whole suite of things. I'm looking at frequency, call rate, inter-call rate, and as well as inter-pulse rate, and other more technical, more kind of properties as well.
Oh, very cool. Yeah, we did some playback stuff with peepers using some of these metrics. There's definitely a good literature out there. I'm looking forward to finding out what you guys find. It's a very cool idea to think of sperm quality and see how honest the call signal is. So yeah, look, everyone's got some really cool stuff this year. Thank you all for sharing.
Amanda?
Thanks, David. I just have a quick question for Barbara. You kind of touched on this, I guess, when you mentioned that some patching was needed to be done after the 2013 to, I guess, 2020. So I was wondering what the lifespan was of some of the fencing that you used, and if you had any comments on that.
Yeah, thanks, Amanda, for the question. Yeah, so we used that white fencing we put at the Lost Shoe Bridge is actually hockey board arena. It's the same stuff they put on the hockey rinks. And it was UV treated. It was supposed to be good for at least 10 years. But we do see on the sunny side of the highway, it's cracking, especially where we bent it over to make the overhanging lip. So we've now started to patch it with metal, like roofing material. And we hope that'll last a bit longer. And we're also kind of starting to think, maybe we shouldn't be putting so much plastic in the environment. What's been really amazing is how well the fabric fencing works. It's a pool fabric. That's what you put over pool covers. The only thing that gets it is when trees fall down on it. So yeah, it is a labor of love keeping these things going. And one other cautionary point about fencing is when the Parks Canada hired a major contractor to do the big project in constructing their bike path, and they ended up subcontracting to these guys that build picket fences for the fencing, the frog fencing part. Ooh, what a nightmare. Everything is being repaired now by one of our local frog fence guys who is taking a lot of time to try and fix some of the mistakes. So if you're ever in a situation where you get to advise on installation of a frog fence, make sure you get somebody who understands how they're supposed to work.
Jonathan, that might be the last one.
Yeah, just to give some attention to Briar a little bit. Question about what is the reasoning, I guess, what would be the reasoning behind thinking that the poor reproductive success in captivity was genetic as opposed to some sort of behavioral issue, like potentially that we're seeing with the chorus frogs?
So we're actually looking at both, because I only did 5 minutes. I only talked about the genetics. That one's a little more straightforward. We're also looking at more behavioral and enclosure conditions based on, because it was so successful this past year, we're doing a whole controlled setup this upcoming breeding season to see if that is more of an effect. Especially because they're very communal breeders, which is very difficult to replicate in captivity. So we're looking at a lot of that. The genetics is more of, we already know that they probably have really low diversity in the wild based on there's a couple studies done on populations in the States that said that they already had really low diversity years ago. And the captive breeding programs right now have mostly been set up to really optimize retaining that genetic diversity instead of moving towards more of a communal breeding, because that would be harder to track genetics, because you're kind of throwing them together more instead of picking and choosing pairs. So we want to confirm more that genetics are not a concern before we move to the communal breeding. So it's more of a checking, making sure it's not an issue, and then we can focus on what might be the bigger issue.
Cool. Thanks for the extra context there.